Hi guys, Miss Francis here. Today I'd like to talk to you about a different type of membrane receptor. It is called the tyrosine kinase receptor. I'm sure most of you know somebody or are related to somebody that has diabetes, whether it be type 1 diabetes or type 2 diabetes. Diabetes is the inability to regulate your blood glucose levels. Now, maybe it's because you don't produce insulin, a hormone required to regulate that blood glucose, like in type 1 diabetes. Or maybe you produce that insulin, but for some reason, your receptors aren't acting the way that they should, such as in type 2 diabetes. Well, the type of receptors that are going to receive that ligand of insulin are the tyrosine kinase receptors, and they're going to regulate and coordinate a variety of activities in addition to regulating blood glucose levels. Where the cytoplasmic side of the receptors functions as a tyrosine kinase, which is ultimately going to take a phosphate group from ATP and add it to tyrosine on this protein. Well, what does this individual tyrosine kinase receptor look like? It has got two binding sites. This is where the ligand will bind. A single alpha helix spanning the membrane, and then a tail that consists of several tyrosines. But do you see how each tyrosine kinase receptor consists of two polypeptides? They are referred to as tyrosine kinase receptor proteins, and in this state, they are inactive monomers. However, when two ligands, two signals, such as insulin, for example, bind to our tyrosine kinase receptor, we form what's called a dimer. And now my tyrosine kinase receptor is activated. However, to get a fully activated tyrosine kinase receptor, I need to phosphorylate my dimer, meaning I need to take a phosphate from ATP and add it to each one of these tyrosines. Now, my fully activated receptor activates those relay proteins, which then triggers many different transduction pathways and responses. Examples of signal molecules that are going to bind to tyrosine kinase receptors are insulin and growth factors. Insulin is the example that we just talked about. So here's another image that depicts the sequence of events that happen when ligands bind to a tyrosine kinase receptor. Now, what's important to realize is that a single tyrosine kinase receptor can elicit two different cellular responses because of the ability of a relay protein to be activated by these different tyrosines. So here's an example of what happens. You've got your receptor and you've got your signal molecule. This is an oversimplification. They're just showing one signal molecule. We know that two signal molecules need to bind to this receptor, which then forms a dimer, which also isn't shown. That will cause this activated relay molecule, which then sets off this phosphorylation cascade, which ultimately leads to some sort of cellular response. So once the signal is received, transduction is initiated, and a series of reactions occurs like falling dominoes. So do you need to know all of these individual steps? Do I need to know that inactive protein kinase 1 becomes activated, and then inactive protein kinase 2 becomes activated? You don't need to know all those individual steps. What you do need to know is that the phosphorylation of the dimer, the 6 ATPs that are phosphorylated, are going to cause an active protein, an active relay protein, which will result in a cellular response. So What do you need to know then? You need to know that ligands, for example, to insulin, will bind to a receptor. And in this case, our receptor is the tyrosine kinase receptor. That receptor changes shape. It becomes a dimer. The dimer is now phosphorylated, and my receptor, my tyrosine kinase receptor, is fully activated. That ATP phosphorylates the dimer, triggering the cascade of reactions, which causes a response. Diabetes also involves a hormone that doesn't really get talked about much. You see, even in this image, insulin is represented, but glucagon isn't mentioned. And don't confuse glucagon with glucose. Glucose is a monosaccharide. Glucagon is a hormone. Both of these hormones are produced in the pancreas. Insulin comes from beta cells, and insulin are going to decrease blood glucose levels. In other words, your cells are going to uptake that glucose, whereas glucagon increases blood glucose levels. Let's say, for example, you haven't eaten in a while, so it's going to enable blood sugar levels to increase. Where this balance of hormones enables homeostasis with respect to those blood sugar levels. So if I was to look at the specific pathway for insulin, there are three overall processes or steps that occur. The first one being reception. Right? So my insulin binds to a tyrosine kinase receptor. Then transduction occurs. The binding of the signal molecule alters the receptor, and then you've got this cascade of reaction that occurs when that dimer is phosphorylated. This GLUT4 containing vesicle is going to move. It's going to translocate to the membrane, and those GLUT4 are going to be embedded into that cell membrane. The GLUT4 is a channel protein, which enables glucose to be reuptook by the cell. This is inside the cell, inside the cytoplasm. So that increased cellular uptake of glucose and an increased production of glycogen from that glucose in the liver and in the muscles. But now what happens to the tyrosine kinase receptors? What happens if the tyrosine kinase receptors are inactivated? Well, diabetes, like we just talked about, Alzheimer's disease, multiple sclerosis, cystic fibrosis, all involve inactive tyrosine kinase receptors. Well, what happens in the opposite case if these tyrosine kinase receptors are overactivated? That's when you get cancer. You get that uncontrolled cell growth. The third type of receptor are the ligand-gated ion channels, which is what we will talk about tomorrow.